Hi, how are you?
Good morning.
Morning.
No picture from you.
What do you mean?
Oh, there you are. Okay.
Oh, yeah.
Okay, I just sent you an email a few minutes ago.
Okay. Sounds good.
Okay, it's about Basilaria.
Oh, okay. Was there new action on that front, or?
There's a new participant.
Okay. We'll have to take a look at it after the meeting.
He's building a robot.
Oh, okay. Was he inspired by the work we've been publishing, or?
Oh, yes.
Oh, okay. That's good. It's good to see someone want to do that. Or something.
Yeah, you might want to rope him in.
Yeah, I'll have to take a look. All right. Yeah, hello, Morgan. How are you?
Never seen the real Morgan. Just the electrode Morgan.
Yeah.
Hello.
Oh, hi.
Oh,
there
you
we go.
is it?
There we go.
Yeah. I'm back in San Francisco.
All right. Yeah.
Just getting. Got to get my daughter ready for her summer camp.
Oh, okay. Yeah.
Yeah.
Susan, hello.
Hi.
Morning.
Hi.
So how are things with you?
I'll take an hour and visit with you guys.
Yeah, sure. That's great.
Take a break from it.
Yeah. All right. So yeah.
Yeah.
It looks like we have Andrii and Jayadratha. Hello.
Oh, are you hear me?
Yeah.
Oh. I just want to drop by because I recently get active on GitHub again.
Okay.
In the reviewing all the projects, let's say. I hope you.
Yeah, that's great. Thank you. Yeah. Welcome. Jayadratha?
Hi.
Hi.
Yeah, hi. After a long time, I'm again joined this meeting.
Okay. Yeah.
I mean, recently I graduated. I was super busy for last few weeks, and now I think I can join every week, maybe.
Okay. Yeah.
This discussion.
Yeah. So yeah, you've been wanting to give an update on your Google Summer of Code work.
Yes.
All right. Yeah. Why don't we do that now?
Okay. So I was working on this, like this data, that is this paper, Connectomes Across Development Principle of Brain for C. elegans, and it had total 8 different snapshots of the brain connectomes. And what I tried to see if we can create some discrete time temporal graph out of it, and then maybe run on some DTDG framework or models on this data. So when I was trying to see and like do some data analysis, I found it has a lot of things to cover, which I didn't have much knowledge about, and these things are, I mean, really cool. So I found there is some data sets that is available in OpenWorm in this website, and it has some cool visualizations, some electrical and chemical. These are the connections that was there. So what I downloaded these data from here from OpenWorm Connectome Toolbox, and it has all this data available here. Somewhere here, yeah. These are total 8 different Excel sheets, and in 8 different Excel sheets, it has these 8 different snapshot of the data. So in these snapshot data, like these are L1. In L1, they have 4 different snapshots, and then L2 there it has one, and L3 there is another, and L4 there is none, and L4 then adult there it has 7 and 8, and they have written it is here somewhere here. Yeah, so all the data set where it has come. I mean, L1 is at birth, the data, and then L2 after 5 hours, and they have taken a snapshot of the connectomes and its connections. And then data set 3 has at 8 hours, and then L1 data set 4, it is at the larval stage, that is estimated to be at 16th hour, and then data set 2, it is at the end of the larval stage, that is at 23rd hour, and then L3 27th hour. It is like adult, and then young adult, and that is at 7, and data set 7 and 8, that is at 45 hour. And so basically what I have done in this week, I just created I started creating this. I downloaded those data at first, and then I tried to see what are the connection it has. So dataset 1, it has this many connections, 858, and then it has 146 presynaptic neurons. These are unique number of presynaptic neurons and 173 number of postsynaptic neurons. And one thing I have seen here as this one postsynaptic, this increased a bit as we go through like 8th dataset, and the number of connections, that is the number of ages. So you can see connection edge number of ages that increased as we go from birth towards adult age. As we have gone, we have seen number of connections that increased here from 858 to 2496. So this is the data that is available. So these are the presynaptic and postsynaptic, and then type, electrical, chemical. These are the type of connections, electrical or chemical, and then synapses. How many connections between these two, presynaptic neuron and postsynaptic neuron, and these are the number of connections. So it signifies the strength, how much strength it has. So this is what I found, and then I analyzed each of the data, like synapse, the number of for each data or each page, like in dataset 1, how many chemical or electrical data that are available, and the count range, how many counts are there. In some of them it has 1, some of them it has 9, some of them it has 8, like that, and the mean synapses per connection. So as we can see here, so suppose here in this connection they have 3 number of connections, here they have 9 number of connections, here they have 6 number of connections, and most of them it has only 1 number of connections. So here I have shown the average mean synapses per connection, that is 1.63, 1.83, like that. So here also you can see at the start, like during the birth phase, the mean synapses per connection, that was 1.63. As we go towards adult phase, it is increasing. So 1.63 to 1.83, then 2.202, and synapses count also, the range, that also increases. And as we go to dataset 8, now it is 3.37. So as we're going towards adult phase, the number of synapses per connection, that also increases. That means it strengthens the connection. And another thing I have seen is the chemical. For chemical, the connection, it is much more than the electrical connection, and it was shown here also. In the paper also they have written somewhere.
Jayadratha, are those connections from one cell to another in both directions or just one direction?
I guess one direction.
One direction.
I guess so, because presynaptic neurons are I mean, it is going from presynaptic to postsynaptic. Is it? I'm not sure about that. Is it? Is it the case?
Yeah,
Presynaptic to postsynaptic.
I think somewhere in that data set they do have a distinction between pre and post. So you can actually get the orientation, but I don't know exactly in this case what yeah, I don't know how that's stated.
Yeah, I have seen like here they have shown the like arrow mark. So they from there I found maybe it is because it is going from P to post. Yeah, I mean
there's
it was as assumption by me.
Yeah, so you'll get a lot of reciprocal connections between cells. They're going from one to the other and then one to the other, and but this is something that in development you're going to see changes in. But yeah, I don't when you get into the data set a little bit more, maybe there's some interesting things you can do with those kind of directional connections and figuring out like how they change. So.
Okay.
I didn't see any two-way arrows in those pictures.
Yeah, that is the thing. I also didn't see much, but it's like mostly it is one-sided. There might be, if we zoom more, maybe we find.
Oh yeah, there's a pair. Yeah, I see a pair there on the left.
Okay, where it is? Right side here, somewhere.
Is this AWC cell have a synapse to itself?
Oh yeah, this one.
Yeah, they can synapse
Yes.
upon themselves.
They can. Okay.
Yeah, I mean, and think about it like cybernetics, where you have feedback loops and you have self-regulation, you have auto-regulation, you have regulation between pairs of cells, a lot of articulation in those kind of networks. So yeah.
Okay,
But
and yeah, here I found one. It is both the connection there that like from ASH to AVH, then AVH to ASH. Here I found some like both the connection.
okay, yeah.
Yeah, this is at the adult data set.
Do these neurons have any distinguishing features other than the lineage tree? Do they look different? Are they chemically different?
I mean, these things, they, like chemically different, I'm not sure, but I guess since they have different name, probably they are like they have different characteristics.
Well, I thought the names came from the lineage tree.
Which names? The names of the cells?
Yes, the names of the cells.
Oh, those are those cells are neurons. So the neurons don't come directly from the lineage tree. That's the adult nomenclature. But this is so AWAAIY. Those are the adult differentiated neurons, and then those differentiated neurons come from the lineage tree, and we have information on like where they come from in the lineage tree. If you go to the interactive lineage tree, which we talked about, that information is in there.
I found.
Yeah, and you have.
I found this website. Yeah, in nematode.org, I found all these connections are there. So suppose if I write about this ADAL, so it has all the connections that are available. So these are chemical synapses and these are gap junctions. Gap junctions, they're telling the electrical as gap junction probably. Like what we have written here, the electrical type. So electrical type is they're showing it as a gap junction. And we can take any, suppose this one, I'm taking this one, and we can see what are which one is connecting to what. So
Yeah, I guess I'm asking is, do these cells look different? They have different shapes?
Yeah, I mean, they're
uh,
different shapes. You mean the morphology of each cell?
Oh, yeah.
Yeah, I think they're different shapes. I'm not really sure. You'd have to go to an atlas to see, but I think they get they're different shapes. They have, and of course they have the gap junction connection, so they're not just spheres, you know.
Okay.
and then like from paper I have taken these age hours when this data set has appeared, so that I can create snapshots at different so like we can create the DTDG, that means discrete time temporal graph data set. So each snapshot will be one graph, and this is what I tried to see. So here, so overall, I have seen the total number of unique neurons that are 225. So that will be my number of nodes in the dataset, and in each node will have connections, and it will be directed graph, and for each snapshot, and the direction and the age strength will be the number of connections it has, or this one, synapses it has. So for this one, it is 1. For this one, we will have 3, like that. And this is it. Till now, I have done this, and now the thing I'll do, I'll create a DTDG, graph out of it, and then I'll see few of the recent methods that are available, and I'll try to model those.
Okay.
Yeah. So, I mean, in this field, I am very new, and understanding these is little difficult for me initially. I was, going to different websites, and also I took help of ChatGPT as well.
Okay. So to be clear on the data sources, you have the data sets from L1 to young adult, and you have these sampling points. And then you have the neurons that are connected, like pairs, and you have the reciprocal connections, so you have one to one, one to the other. Like this is typical of connectome, except you have time points in development where there's synaptic information instead of just the pair connection information. And then you're going to have this basically you can build directed graphs from that and build an evolving graph essentially from L1 to L.
Exactly.
Yeah.
Yeah, exactly. That is what I want to do. Like create this graph and then try to see if there is any intermediate step we can predict, something like that.
Yeah. So yeah, it's interesting in this paper they go through some analysis of the different counts over time, and like I actually wrote a paper where I used the data set as well, where you do the counts over time and you get these fluctuations, and it's interesting, but it tells you a little bit about like what's happening with synapses at different points, different circuits. So you could take like a subset of the adult cells and say these, or I guess terminally differentiated cells, and say that this is what's happening in the connectome during larval development. And there should be a lot of plasticity. There should be a lot of fluctuation between the synapses, and they get pruned and they come back, and depending on what's happening. And this is only taken from one worm. So sometimes worms will suffer things like starvation, or they'll suffer resource deprivation, and they'll do things like go into developmental arrest, or they'll go through this dour stage where they have these neuroprotective adaptations that they take on. So these are all things that kind of are interesting biologically. But this is like a nice temporal graph because you can get a sense of like with the network, how the network changes, just, what the fluctuations are, maybe find some interesting things with respect to some of the circuits, like some of the movement circuits. We can isolate that and say, hey, the movement circuit is changing in this way during development. And, there are these synapses that are changing. And we can also model the synapses once we build the temporal graph to say, okay, this is maybe how it's affecting behavior. So I think,
Okay, yeah.
yeah. And then the other thing is there are these stable connections, and then there are these, I know they have like a typology of connections. So they have the stable connections, which are usually things that persist across development. They have like the developmental connections, which are like, in development, you get special connections just to deal with what's going on in larval development. And then these like transient connections. So those would be interesting to break out as well. Like you build a series of graphs with just one set, a series of graphs with another set, and so forth. So, yeah, I think you have a good baseline though here for like building these. So like you have the data, you have a method for getting it into Python and playing with it and building the graph visualizations. Yeah.
Yeah, definitely. Okay.
So your plans for next steps for that?
Yeah. So as I told, plan is to create this graph out of it. And so till now, I found the number of nodes and the connections. So those things are already available. And the edges, the edge, that means who is connecting to whom. And now I'll create a data set, that is temporal data set. And then I'll try to find through using some already existing temporal graph models, and we'll try to model it through if we can find some exciting pattern in between. Suppose in L4 stage there is no data set that is available, so can we predict something in the L4 stage? What is the connection that can be there? So that is what I wanted to do.
Okay, so you want to make like basically the visualizations, but also predictions to L4. So like maybe between
Exactly.
developmental stages. Yeah. Yeah, I think that's good.
So So all these things till this point, till data set 6, I'll create a visualization and see what are the, how it looks like, the connection looks like. And then using these connections that are already available, can we predict something in between or intermediate steps? So that is what the goal is.
Okay, Jayadratha, do you ever get connections between cells that are far apart?
I didn't get it. Can you please repeat?
I think he knows that yet. Well, yeah, that brings up another question, which is, can we like find the developmental precursors to these adult neurons, and then can we find the positions from the developmental data that we have? So we have a developmental data set that has the position, like from cell tracking, has the positions of the developmental cells, and then you can figure out where the neurons are. Actually, you can figure out where the neurons are from like another atlas too. But this would be, kind of thinking about where those neurons are in the nervous system. Yeah.
Oh, okay. So
okay. So 3D connection, you're telling.
Yeah, some sort of 3D
actual
position.
point.
Yeah.
So we don't know at this point.
No, we don't know. In this data set, there is we don't have that.
Okay, so those graphs do not reflect distance.
No.
No.
I'm wondering if they're. Oh, go ahead.
Okay, so you'd have to accumulate the data of where each cell is and then calculate the distance for each of those pairs.
Yeah. Yeah, I mean, if we have that the 3D like where it is and its 3D coordinate, then getting the distance, that is very easy.
Okay, I guess when two cells synapse, the question is, do they only synapse to local cells, or do they reach out for cells that are far away?
Okay, Andrii, would you like to say something?
Yeah, I remember then when I look at visualization of the Sirihanis, for example, and I noticed that a lot of cells which are very, very long and almost that's muscular cells probably, which very long and mostly like they connect different parts like head of worm and the tail of worm almost. It's like it's not all of them, but it's it's hard to guess, and all they like not cellular, but like not plain, but some kind of like cake when you press something, and just something like that. I mean, I'm not sure.
Yeah.
It's hard to generalize. I think distance from such shapes. It's like it's probably if we somehow divide by morphological properties, maybe it's like only that makes sense, because some of them large, some of them compact, etc. No, kind of 3-4 different kind of shapes.
Okay, in other words, the centroid of a cell may not have much meaning for a cell that's elongated.
Yeah.
Yeah. Okay.
Yeah, you have a lot of bilateral pairs too, so you'd have to count, like, there'd be a longer distance, but it's like, well, it's a bilateral pair. So there's a functional reason why. But yeah, I mean, if we could get distance data, it would be interesting. Certainly, like if we could say, these are head neurons, these are tail neurons, like what is going on there, those connections. Because that's a functional hypothesis. That's like, okay, well, something's going from head to tail. There's, obviously some function to that. So yeah, we could say things like that. I mean, I think the biggest thing though right now is just getting the model working, like getting it in place, generating the visualizations, and seeing if we can kind of make some pictures.
Bradley, maybe distance between cells, distance between their membranes rather than their centroids.
Yeah, we could do that.
If these data are available somewhere, I mean, like people who have collected this data, if they have taken while collecting, maybe it was there. Otherwise, I guess it's difficult, right, to get.
Well, there might be some connectome cell tracking data. We could also take it from the OpenWorm browser. I'm not sure if those data are, like if we have a quantitative, we should have it, I don't know, somewhere. But we can look into that, see where we can get those data.
Okay.
Anyways, I think that's good. That's a good update, Jayadratha. Thank you. So yeah, keep on working on this. This is great. Yeah.
Sure. I'm working on it.
Yeah.
Yeah, actually, I mean, I'm very new into this type of data, and the terms, like terms that are there. So to understand those, it was taking time.
Yeah.
But otherwise, development is not that difficult for me. But understanding the data and how can I contribute there, that was taking time.
Bradley, there's also the question of whether or not the synapses are mosaic, the way the cells are.
Okay.
For example, if you take a left-right pair, of cells that synapse each other, do they also synapse corresponding cells on left and right of the embryo, the animal?
Yeah, I don't know the answer to that.
In other words, is bilateral symmetry preserved or not?
Well, I think a lot of the bilaterally symmetric cells are synapse each other because they have like a functional animal.
But I'm asking about their synapses to other cells. Are they the same? Is the
Oh.
left cell, the right cell, the same?
I'm not sure. Yeah, we could find that out, I guess.
Yeah, so that would indicate whether or not the synapsing is part of the mosaicism.
Yeah. We'll have to look at that. Yeah, it might be an interesting question. All right. Yeah. All right. Thank you, Jayadratha. That's good. I see Hussein's here. Hello, Hussain.
Thanks. Hello, nice to see you guys.
You wanted to talk a little bit maybe about what you're doing these days?
Sure, yeah, a few things. Yeah, thanks so much for this. So Dick and I have been working with a group in Italy with Sabina Longa on our G-Flow peptide membrane product. Okay. Excuse me. So basically, we are now in the process of applying for a grant. We're in the process of applying for a grant to potentially fund research on peptide membrane dynamics as it relates to the origin of life. Yeah. Excuse me. Yeah. I don't have so much else to show and present right now. I can show some of the images from the report. I think that might be okay. A statistical mechanical model for lipids first hypothesis. Oops. Excuse me. We are drafting a bunch of papers, and we're drafting a bunch of different things. Excuse me, on simulations to determine how peptides, how the lipid membrane, to study and determine some of the mechanisms underlying lipid formation, the lipid membrane formation, particularly in the context of peptide rafts. And with the hexagonal grid model, we can simulate the lipid membranes and peptides and how they evolve under very simple physical constraints. And together with Savino's group over in Italy, we are applying for some grants to get this together. That's basically all I have to share right now. I can share some of the outputs from our just very simple simulations.
Yeah.
Basically, we're trying to simulate affinity dynamics. Excuse me. So we introduced these hexagonal grids. All right. So basically, when we're talking about the origin of life, how did cells come about? How did life come about? We answer questions of nucleic acid replication, and I think there's been a lot of evidence showing that the peptide interactions, they themselves preceded the genetics. So peptide alpha helices in particular, we can embed them in membranes based off the hydrophobic thickness and create stable microdomains that influence membrane behavior. And we can simulate those with these hexagonal grids, these hexagonal, we call it a chevron pattern. I think that's what it's called, yeah. Chevron. So this is a chevron pattern when things are just arranged in a V like that, yeah?
Yeah.
We can simulate these hexagonal grid lattice structures in a simplified lattice system in which these hexagons these hexagons form a grid that show how lipids or peptide units bond with one another. Okay. Yeah, and we're writing some papers and drafting some things together to hopefully apply for a small scale grant. I think that's what I mentioned to you in Slack. That's what I have to present right now. Yeah.
What about those? Did you have those visualizations, or I saw those graphs that you were showing?
Yeah. Some random graphs
just to see those.
that simulate binding affinity over time. So each of these visualizations represents. Let me share my screen again. So you can
Yeah.
These visualizations represent how peptides might move in space and bind over time. Now this is all completely random because we just want to show that we could move them all together. But yeah, the model, so the hexagonal lattice, excuse me. The hexagonal lattice is meant to simulate the peptide dynamics themselves. Yeah.
Okay.
We want to simulate how they can interact with the lipids and drive early membrane structuring and clustering the vesicle dynamics themselves. So this is all completely random, but it does show that we can get them to move over time and obey very simple rules or mechanisms when, say, affinity turns off or affinity turns on, or similar simple things. Yeah. So cool stuff. I think it's cool.
Great. That looks great. Yeah, I'm looking forward to seeing the outcomes of that.
You guys ever played Catan? That's what it reminds me of.
Oh yeah.
Settlers of Catan. It's
Catan,
a
the
board game
game.
with
Okay.
these hexagon board game units. That's always what I think of. Settlers of Catan. With that.
Yeah.
Is it like Chinese checkers works too?
I guess Chinese checkers is fun. That's a hex, right? The Chinese checkers, right?
Yeah, I think so. It's been a long time since I played it.
Yeah, there it is.
So it looks like the center is a hexagon, but it's actually 6 triangles together. So that would be a hexagon. Yeah.
Yeah.
Cool. Good stuff. So yeah, and the chevron pattern. That's what I found when I looked this up here. Yeah. The chevron is the name of the pattern. So cool stuff.
So where does the chevron pattern fit into that? I didn't notice.
Oh right. So the chevron pattern. Dick had a better image. Let me show up the better image that I had. The chevron pattern. Well, the chevron is the stripes in a line like that. That's the chevron pattern. It's not specific to this. Yeah. Excuse me.
All right. So this is supposed to be like early pattern formation or something like that.
Yeah. So the other we are just showing that we can move them right now in Python. That's the important thing right now. Okay.
Yeah.
Then we can code any sort of simulation rules or methods or mechanisms for the research itself, and we've been basically devouring tons of papers, like the gluttons that we are, and we want to consuming a lot of these. Do you like there was a Deemer paper that Dick had given. Excuse me. I'm like jumping from many different things from one thing to another. Right. The Deemer, like for example, we can start with this one, right? This paper from 1991 looks like it was from 1991. Okay. Polycyclic aromatic hydrocarbons, primitive, like in this one, for example, yeah, it looks like this one they're proposing the very specific hypothesis of how, excuse me, how polycyclic aromatic hydrocarbons (PAH) would absorb light in the near UV and blue wavelengths and are candidates for primitive pigments. Like that's one example of how the origin of cellular life may have come about. And the papers themselves are pretty interesting too, or the figures are pretty interesting too, excuse me. Say how you can go from, because they describe the hexadecane purine mixture in this one itself, yeah?
Yeah.
I thought that was interesting. Dick, do you want to talk more about this one, the Deemer? The Deemer paper, excuse me.
Yeah, I've been looking into these. They're abbreviated PAH.
Right.
Yeah. Cool.
Aromatic hydrocarbons. Now, these things are usually quite poisonous. But one thing which Deemer didn't emphasize is that because they're hydrophobic, they concentrate in the membranes. And therefore, the membranes can actually protect the rest of the cell from any PAHs on the outside. And meteorites are full of them. So they're probably in there. I've also been finding that the PAHs can actually make the membranes swell a little bit and increase the permeability. Because one of the problems that early life didn't have to solve is that small molecules will go in and out of a vesicle.
Right.
Okay. Yeah, I think turning the hypotheses on its heads and seeing just opening it up for question and do these sorts of simulations. That's a very good way of starting. Okay, here we go. I found the image that I was looking for. The triads, triads. So not just hexagons. Excuse me. Not just hexagons. I grew up in a house where we speak like 4 different languages, and all my family is from different countries. So my pronunciations of things will change within their own sentences. So with English words, stop. But we can arrange. Like my family's from UK. We also date back to India, and we speak like different languages and even accents between my American pronunciations and my family's British pronunciations. They just get mixed up all the time. So, excuse me. But you guys see these walls. This is the chevron structure. The chevron pattern. The chevron pattern is just like kind of like stripes arranged together. But we can form walls. We can form walls with these things. Yep. And this is what it would look like. If you wanted to simulate the peptide pattern. This would be what we would be looking for. This would be one of the types of patterns that we'd be testing how this could come about in some ways. I think Dick had showed me one written in one of his manuscripts. I think so. Like because you showed it was like made with like MATLAB, right? Yeah. Okay. But yeah, cool stuff. That's what we are working.
Yeah.
Are there any other tilings you could use other than the hexagon?
Okay. Well, in theory, obviously, yes. But see, the question is that the choice of using the hexagon matching the structure of the lipid peptide membrane versus other ones, say like even a square or a pentagon. I think in many ways you'd ask yourself what sort of physical constraints on the system you would have to impose, and like for the PAH, for example, they'd be typically shown as derivatives from like hexadecane, say. So if we wanted to get more fine tuned, perhaps try even like a pentagon, and what sort of geometric formations might come about with that one. Like we definitely could, but not right now.
You'd have to combine a 5 and 7 sided geometry in order to get a tiling.
Yeah, Susan, we're making a simple approximation.
Oh, okay. And it's also molecules, so they're more rigid.
Yeah, basically,
the stability hypotheses for those sorts of.
A lipid is represented by one hexagon. The alpha helix of an embedded peptide is represented by the 3. And that's having to do with the ratio is approximately 1:3 for the widths of these molecules in the membrane. So we're doing a crude approximation to avoid the problem of having heptagons and pentagons and in there, and we're assuming a planar membrane.
Okay. Yeah.
Okay, so it's a rough approximation.
Yeah. Okay. Yeah. You're modeling it. It's okay. It's okay. I just thought I'd ask.
Yeah. I mean, yeah. So cool stuff.
Yeah, Susan, to put it another way, most Ising lattice simulations are done on regular grids. And we suspect the membrane is acting somewhat like an Ising lattice and giving some phase transitions. Okay. Now, yes, it could be made more complicated by having an irregular grid. Okay. But then every part of the grid would have to be simulated separately.
Hexagonal structures just give so much of that strong balance between a lot of uniformity. All the pieces just arrange them with hexagons that can connect and even intersect. but a lot of flexibility even with those things too. So
Yeah, they're easier to work with. I know this.
There's a lot less programming.
I just went through a bunch of modeling using hexagons. Yes,
yeah,
they're easier to work with, and they're supposed to be more stable.
yeah, on that level, of course. So cool stuff. Yeah, this is all exciting, and anything that can be simulated through the programming, we pose questions on. So
Okay.
All right. Well, thank you, Hussain, for that update. Looking forward to seeing it move along. Yeah, looks like.
Yeah. Sorry, just for context, we're applying for a grant, the Space Exploration Grant Program through I think it was called Chiwi. That's what it was called. It's not a name for it. It's CHIWI Foundation for Space Exploration Science Grants like this one. Yeah. So that's the context behind all this. We're trying to get money.
I'm trying to get money.
Trying to get money.
No, it isn't.
We're trying to get money so we can do groundbreaking research. Yeah.
So we can eat.
Yeah.
That doesn't hurt. Okay, but yeah, cool stuff.
Yeah.
Okay.
All right.
Yeah.
Well, thank you. Yeah. So I'm going to present something here for the last 10 minutes. It looks like Lalith is having connection issues. I was going to ask him if he had something, but we can catch up later on that. And he's given a nice set of updates in our Friday meetings. So we've got that down. But let me move on to this article. This was posted in the Slack by Jesse Parent. And this is from the Wolfram community. So this is kind of like their community site. And of course, the Wolfram community, they're all interested in cellular automata and sort of these evolutionary cellular automata, using cellular automata for everything and evolving them in these arrays. So what they're doing here is they're simulating, well, they call it biological evolution in the Wolfram evolution model. So Wolfram has an evolution model that's based on cellular automata and these propagating patterns that come down like this. And the question is, what shapes can organisms have? And so, this is an idea where you want to get an account of all possible shapes an organism can have, and then, understand sort of the constraints to those shapes, and then understand sort of the number of pathways evolution could have taken to form shapes, to form body plans, and to form phenotypes. So this is basically the cellular automata, and because the cellular automata are computationally irreducible, it's hard to predict the next step in a cellular automata. There's very much the stochastic component to their evolution. So you can see that, basically these cellular automata are propagating according to these different parameter values, and they're using this as a method. They're using this as an analog for the generation of phenotypes in biological evolution. So they say due to its computational irreducibility, predicting the behavior of cellular automata is a difficult, almost impossible task. However, can we introduce the behavior of a cellular automata? Can we induce the behavior of a cellular automaton? More specifically, can we control its shape? In order to answer these questions, an experiment was conducted to evolve various cellular automata configurations to a specific target shape, considering cellular automata boundary as its fitness function. I'd add to this that there have been a lot of attempts at this to sort of get to get your arms around sort of the development and evolution of shape. A number of groups have done papers where they've used cellular automata to do this, and they always kind of come up with this method where they're kind of imposing some sort of template on the shape, and maybe that's not how nature does that. So that's just
Is that what the dotted line is?
Yeah, this is sort of the dotted line. This is sort of the boundary of this region. So yeah, it's they're always trying to do this sort of impose these sort of top-down mechanisms for controlling shape, although you generate within that boundary. And, there's no evidence that nature really does that. So that's kind of the caveat here. So what they do here is they talk about analyzing different configurations. So they generate these configurations. You see this here. This is the density plot of the cellular automata. This is the output step, and this is like the width of it. And then they do this frequency plot where they analyze the frequency of different phenotypes and their rank in terms of abundance.
Bradley, I can think of a case which needs investigation.
Okay.
If you grow up a chick, it has a very rigid shell.
Yeah.
And therefore, the shell may constrain the shape.
Yeah.
And the question is that correct? In other words, if you could grow a chicken without a shell, would you get a chicken?
Right.
Okay, not obvious, but it's an example of a rigid constraint.
Right. Yeah, I think there are some early experiments in C. elegans where they took the C. elegans embryo and they split it up into two parts. So they split the anterior end and the posterior end, and they kind of let them develop separately, and they developed these weird shapes. So they didn't develop according to the sort of.
Oh, really?
Yeah, yeah, I don't know. It's like the cell lineage kind of developed independently, and they developed these barbell shapes and things like that. So it wasn't really constrained by. Well, some people would say it's maybe the boundary of the space. Maybe it's some sort of signaling that goes on between cells. So it could be chemical signaling or electrical signaling that kind of holds that shape together.
Interesting.
But
we did an experiment on axolotls where we constrained the axolotl to the developing axolotl to be between two glass plates.
Okay.
And we ended up with what's called, I think it's called kurtosis. In other words, the back was arched, so the head and the tail were opposite each other, looking out rather than straight.
Yeah.
But that's an example of an artificial constraint.
Yeah. Yeah.
Our skulls grow within constraints. Yes, if the constraints on a skull and an infant are constrained because the soft spots, the fontanelles, are connected wrongly, you'll get an odd shaped skull.
Yeah.
So that's a mechanical system. Also intention.
Yeah.
So.
I mean, there are these examples of like kind of boundary conditions, and then there's like kind of this, and then you have local things, and then you have this global sort of constraint. So it's hard to say which things are responsible, if they're all responsible. But anyways, this is, I think, an interesting exercise in sort of analyzing sort of the evolution of shapes using a cellular automata. So if you generated a bunch of possible shapes, what basically shows up, and what is their frequency? So you might get things that are more common than others. There's this idea of carcinization in evolution, where if you look at a phylogeny of different types of invertebrates, they always tend towards this crab phenotype. So it has like this. If you know what crabs are, it always has that same phenotype where it has like the center shell and then the appendages, and that happens. It's like that's evolved convergently across marine invertebrates. So you get this idea that there are certain phenotypes that recur, there are certain phenotypes that are common, and then why is that? Is it because they're constrained by some sort of body plan, or is it because that's just kind of a more efficient shape? And so in this case, you get a more efficient shape where you get this, some of the possible shapes are limited with certain configurations. There's a lack of shape variety. Configurations of two colors are not used in the evolution process, so that kind of constrain this further. So this is adapting to shape. So this is the sort of the take-home message here. So some successful shapes for a lifetime up to 50 steps, which is very short, are these types of shapes. So you have this long hexagon, you have this diamond shape, you have this triangular shape, you have this sort of hexagon on top of another hexagon, sort of like a club shape, and then you get these kind of trihexagon shapes that kind of look almost like an organism with a tail. And so this is basically, these aren't, some of these have achieved a high fitness level, but not all of them. These are kind of like the most common outputs. Other shapes were considered, but they had a lower fitness rate, so they were discarded. One of these is this sort of, it almost looks like a clamshell or a hexagonal clamshell.
What is the definition of fitness in this case?
I don't know what their definition of fit. They define it here. Let's see. There's some sort of fit. Okay. Oh, the cellular automata boundary is the fitness function. So it's this boundary that's the fitness function, and it's kind of enforcing. Yeah, basically. Okay.
So there's is that from like from Conway's Game of Life?
What do you mean, the method or the
fitness function?
Oh, I don't think so. I think Conway's Game of Life is on a free grid, so there are no boundaries that they worry about. It's just okay. So then this is the evolution of the cellular automata was made using the adaptive cellular automaton function. This function implements a biological evolution algorithm with some parameters to control the evolution process. They include the cellular automata rule configuration, the mutation rule, the fitness function as we discussed, and the number of mutations made. So they're mutating these shapes. They're imposing this fitness function. They have this rule, so the system evolves according to some rule. And this goes back to the cellular automata rules. So it's basically how each cell interacts with its neighbors. And they list these here. To select the most successful case, this adaptive cellular automaton function was executed multiple times using the parallel table resource. With these results, it is possible to select the most fit and understand the overall behavior of the cellular automata configuration in relation to the fitness shape. So they go through about 100 evolutions. They analyze these for each shape and number of states, and they show these graphs that show kind of the output for these attempts for the 6 vertex shape. So this is the 6 vertex shape changing its shape over time, and you get these slight variations in it. And then, so yeah, the fitness function is shown in blue, so it's this constraint, which is kind of like the top of a hexagon, and then the boundary of the cellular automaton is shown in yellow. So there's some variation in the edge. It overlaps the boundary. Sometimes it gets within the boundary, and then that's kind of like how it's the fitness is assessed that way. So things that exceed the boundary have a lower fitness than things that kind of follow the boundary. And so we can visualize this by plotting the cellular automata at the end of each evolution. So you can see that there are a lot of sort of convergent shapes here for different parameter values. And then, you can also visualize the evolution breakthroughs, and that's defined as when the mutations lead to a greater fitness value for each case. And then this is the evolution of the best case on the simulation example. So you see the evolution here, where these things are kind of assembling here and making breakthroughs as they're evolving. So sometimes they make changes in shape that are unique, that are innovations, and sometimes they kind of follow the same pattern.
Okay, Bradley, is Wolfram himself a co-author on this?
This is just using this environment, so no, he's not. But, he's kind of supervises a lot of the work on this. So one way to visualize this function is by plotting a graph of its evolution. So we can plot out this phylogeny, and we can show that the green nodes with higher fitness sort of predominate down towards the bottom of the phylogeny. So that's right there. And so that's pretty much that paper. I just wanted to go over that to show some examples of cellular automata evolution and shape formation.
Cool stuff. Cool stuff. Yeah. Excuse me. Cool stuff. I appreciate them scouting through different shape possibilities. I hadn't considered that so much before. I figured when studying the origin of life, studying similar questions down there, you'd kind of have to limit yourself to a particular shape, a particular structure from the get-go. So that's really fascinating, especially with cells. The biggest thing that always comes to my mind, especially back when I used to do neuroscience research on, say, like neuronal cells and neuron cells, was the minimum limit for how small they could particularly be, given the complexity of what they contain, especially in particular. At some point you're bounded by all of the all the stuff, all the things inside a cell about how small they can truly be. So yeah, interesting stuff.
All right. All right, so I think that's it for today. Thank you for attending. Andrii?
Yeah, I have a question. So I tried to update the tracker database, which seems to be like neglected for some time.
Yeah.
At least what was suggested on the GitHub. And the one issue is that there are database it by itself. I get lost, or somebody should contact previous researcher, and maybe they have that database. So
Okay.
my question: did somebody try to contact them, or just I have to do that myself, or it's already contacted and nobody knows, or whatever?
I'm not sure. Yeah, you could open an issue on GitHub, but there are also.
Oh no, it already exists.
Okay,
Issue.
it is. Okay. Well, maybe I can get in touch with some of the people involved in that and see. This is the movement tracking database or the movement database. Okay, why don't you send me a message in Slack if you can, and we'll talk about that offline. All right, thank you for attending. See you next week.